All right, we've been talking about our microfilaments, started exploring them. So let's continue our discussion of microfilaments. Actin can bundle together to form microvilli, and these are the best studied examples of actin. Microvilli are important in intestinal mucosal cells, where they help increase the surface area of the cells by, creating these little bumps that go up and down. The core of a microvillus is a tight bundle of microfilaments with the plus ended pointed towards the tip. The microfilaments in microvilli are connected to the plasma membrane by myosin 1 and calmodulin, which will help cross-link that microfilament. And the microfilaments in the bundle are going to be bound together by the cross-linking proteins fibrin and villin. So here are our little microvilli extending from the cell, allowing it to have a much greater surface area than if it just was straight across, allowing it to absorb more nutrients from the foods traveling through the lumen here. So at the base of the microvillus, the microfilaments will bundle into a network called a terminal web. And these filaments of the terminal web are mainly made of myosin 2 and spectrin. And these proteins, myosin 2 and spectrin, will connect the microfilaments to each other. It also connects the microfilaments to proteins in the membrane and might also help connect those microfilaments to intermediate filaments. Microfilaments are connected to the plasma membrane, and they're important during cytokinesis. They help move that cell membrane during cytokinesis to allow the cell to split into two cells. But this connection to the cytoplasmic membrane is indirect. It requires some linking proteins like band 4.1, ezrin, rhodixin, myosin, spectrin, and chirin. Okay, I'm sorry these were misspelled here. Please refer here for the spelling. Actin can also form a dendritic network. The word dendritic means tree-like, so think of like a dendrite extending. So actin helps form that dendritic network by complexing with proteins like ARP2/3 complex, which nucleates new branches to create that more branched tree. The ARP2/3 branching is going to be activated by proteins that includes the WASP and WAVE scar. WASP stands for Wiskott-Aldrich syndrome protein. Sometimes we don't need branched filaments, though. We need long actin filaments. So to create these longer actin filaments, actin polymerization will be independent of the 2,3 complex. And instead it will use proteins called formins. Formins help create that longer actin filament as they're growing along the filament and promoting polymerization and not branching. Both plasma membrane lipids and regulatory proteins can affect microfilaments. They can affect how they're being formed, their stability, and their breakdown. One example is PIP2 that we discuss in our cell signaling and I think our cancer chapter a lot. Phosphatidylinosytol 4,5-bisphosphate. Your textbook also discussed some other regulatory proteins that you are free to explore. When a cell is exposed to growth factors, their cytoskeleton can be dramatically changed as the cell is going through processes now that will allow it to grow. A lot of these signals will affect phospholipids, but they can also affect monomeric G proteins called Rho GTPases. This includes Rho, RAC, and CD42. So these are 3 different signaling proteins that will have different effects on your cytoskeleton. So here you can see in the presence of Rho, you have more of these stress fibers. When RAC is activated, you have more of this lamellipodia. And here when CDC42, you can see these phyllopods, these little fingers being exerted, being released from the cell, compared to a serum starved or a cell grown without any of these growth factors. Okay, so activation of the Rho pathway results in stress fibers, RAC activation results in lamellipodia, and CDC42 in phyllopodia. All right, that concludes our discussion of microfilaments, but we still have one more cytoskeletal protein, our intermediate filaments, to discuss. So I'll see you soon. Bye.